Okay. Good morning, students. I welcome you all back to my next video lecture, online class lecture on leaf curl disease in case of tomatoes. So in my previous classes, we have studied about some fungal disease as well as some bacterial disease and some of the mycoplasmal diseases. So today, let us study about the viral diseases caused in plants. According to your Bangalore University syllabus, BCU syllabus, we are going to study about leaf curl disease in case of tomatoes. As the name of this disease indicates, you can expect or the symptom of this particular disease is curling of the leaves of tomatoes. So that is the reason this particular disease is called as leaf curl disease. It's also called as tomato yellow leaf curl disease because once the leaf gets curled, that leaf also show chlorosis. That is the reason. So this particular disease, as I had mentioned, can be observed in case of tomatoes. So the botanical name of tomato, Lycopersicon esculatum. And the pathogen which is going to infect this particular plant and cause this particular disease is tomato yellow leaf curl virus. It's tomato yellow leaf curl virus. In this particular video lecture, we are going to study about this tomato yellow leaf curl virus, about its structure and its morphology and its genome. And also we are going to study about how actually this particular virus replicates, what is the life cycle, how actually it replicates inside the host, what is the life cycle, how actually the life cycle of this particular virus is, and how actually this particular virus is being transmitted from one plant to another plant, and which is the vector which cause this particular disease. And we are going to study about the symptoms of the disease and the management of this particular disease. So we are going to study about the virus, morphology of this virus, replication of the virus, transmission of the virus, symptom of this particular disease caused by this particular virus, and the management of this particular disease or the control measures of this disease. So let us start with the study of this particular virus. This is one of a virus. This is one of the virus, that is tomato in general, tomato yellow leaf curl virus. It belongs to the, according to the classification, it has been placed under the family Geminiviridae. It has been placed under the family Geminiviridae. What is the reason of placing this particular virus? That is important, students, for the point of your examination. Why it has been placed under Geminiviridae? All the viruses, I mean, you know, the virus has been classified basically based on the genetic material. You know, viruses are made up of what? Protein and a genetic material, isn't it? Genetic material can be either a DNA or RNA. So based on that, it has been classified. And the reason for placing this particular virus under Geminiviridae is because of the presence of a DNA as a genetic material, and the kind of the DNA, it is single-stranded. Students, kindly note, it has a DNA which is single-stranded and circular DNA. That is very much important. You have to know about this. For the point of your competitive examination, they may ask, Geminiviridae, this particular this particular virus has been placed under Geminiviridae because of the presence of a genetic material, DNA, and the kind of DNA is single-stranded. It is single-stranded circular DNA that you have to remember. And this Geminiviridae, this particular family, has got around 485 species. Kindly note down this particular Geminiviridae. It has, in this particular family, there are around 485 species has been discovered till date. There are around 485 species, and most of these particular species has an ability of infecting plants. Most of them has an ability of infecting plants. Another important thing, out of this 485, this 485 species has been, they are under 9 different genera. So, in this particular family, this is Geminiviridae, there are around 9 genera. Or under this 9 genera, there are around 485 species that you have to remember. And the virus which we are studying today, this belongs to the genus Begomovirus. It belongs to the genus Begomovirus, and this is one of a virus has an ability of infecting the plants, especially the plants belonging to the family Solanaceae. Students, kindly remember, they have an ability of infecting the plants belonging to the family Solanaceae, that is brinjal, chilies, tobacco, tomato, potato. All these are the plants which belongs to the family Solanaceae. So this virus has an ability of infecting this particular plants, and also especially in case of tomato, it causes a disease called as yellow leaf curl disease. And this particular virus has been also named as TYLCV. What is this TYLCV? This is tomato yellow leaf curl virus. And as I was mentioning, this is one of a DNA virus. That is, it is the genetic material in this particular virus is DNA. And as I was mentioning, it belongs to the genus Begomovirus under the family Geminiviridae. Students, kindly remember this particular sentence. Very important for the point of your competitive examination. Now, let us study how actually this virus is. This virus is non-enveloped and it is around 38 nm. This is around 38 nm in its length. It's around 38. This is the diagrammatic representation of this particular virus, and this is electron microscopic observation. So this is around 38 nm nanometers in its length and around 22 nm in its diameter. And it is twinned. Why it is twinned? That is, you know, they are made up of what? Protein and what? The nucleic acid, isn't it? Protein forms a protective covering. That is, it's called as capsid. They are called as capsid. So here, the capsid are twinned or geminate. That is the reason it is the family has been named as Geminiviridae. Why it is named as Geminiviridae? Because the capsid, the proteinaceous capsid, is twinned together. It is twinned and it is incomplete icosahedral symmetry. And capsid consists of around 22 pentameric capsomeres. How many capsomeres it has got? 22 pentameric capsomere, and it is made up of 110 capsid proteins. Students, kindly remember how many capsid proteins are there. It has got around 110 capsid protein, and this particular geminate capsid, geminate means what? Twinned capsid. The green color represents the capsid, the outermost covering made up of protein, that is called as CP protein, that is capsid proteins. This capsid protein contain or it forms a protective covering around a circular single-stranded DNA. And this single-stranded DNA is around 2787 nucleobase. It is made up of 2787 nucleotides or nucleobase. Understood, students? So this is one of a virus where the genetic material is DNA and it is single-stranded circular DNA, and this particular DNA is being covered over by a protective covering made up of capsid proteins. This capsid is twinned or geminate together, and it is incomplete icosahedral symmetry, and it has got 22 capsomeres and 110 capsid proteins it has been made. And another important thing, this particular virus has been distributed or found all over the world, both in tropical as well as in subtropical region. And wherever the tomatoes have been grown, if proper measures have been not taken, this particular infection with this particular virus can happen. However, this particular virus was first discovered in Israel in 1930. And now this particular virus has been widely spread all over the world. So students, I think you got it. That it belongs to the family Geminiviridae because of its the nucleic acid, that is the DNA, single-stranded circular DNA, and it is geminate because the capsid protein or the capsid, the protective covering, which is made up of proteins, they are actually twinned together that is geminated together. Hence, it has been placed under Gemini, and the family also recorded as Geminiviridae. You have to remember this many a times. This particular question has been asked in your competitive examination. So now, you studied about this particular virus, how actually this virus is, what it is made up of, and how actually it is, the morphology we have studied. Now, let us study how actually this particular virus transmits from one plant to another plant. The transmission of this virus happen through an insect vector. The insect vector, that is this particular insect, it belongs to the order Hemiptera. Hemiptera under Arthropoda. You know Arthropoda. All insects belongs to the phylum Arthropoda. Under that, this particular insect belongs to Hemiptera order, and it is one of a white fly because of the color also. It has been called as white fly. And the scientific name, students kindly remember the scientific name of the vector which transmits the virus, that is tomato leaf curl virus from one plant to another, that is Babesia tubaci or tubaci. This is the scientific name. Kindly note down, students. They are commonly called as white flies. They are also called as silver leaf white fly. They are also called as silver leaf white fly or sweet potato leaf white fly. So, kindly note down the scientific name of these white fly which transmit this particular virus, that is Babesia tubaci or Babesia tubaci, and it is commonly called as what, students? Silver leaf white flies, and it belongs to the order Hemiptera under the phylum Arthropoda. As you all know, this is one of a vector where the primary host is tomato plant. Tomato plant is a primary host for this particular virus, and also this virus and also this particular vector can also infect the other plants belongs to the family Solanaceae. Say for example, eggplants, brinjals. Even onto the brinjals, you can see the infection through this white flies. Tomatoes, tobaccos, beans, peppers, all these are also some of the other plant host. However, the primary host is our tomato. Even other solanaceous members can also be the reservoirs of this particular virus, because this white flies has an ability of infecting those solanaceous member. And also recent study also demonstrate that this particular virus can be transmitted to the offspring. Say for example, this white fly, if it has been infected or if it carries the virus inside it, this particular virus can be transmitted even to the offsprings of this particular white flies for 2 generations. And also various studies also demonstrates that this particular virus is sexually transmitted from one insect to another insect. So, so these white flies, they are the scientific name is Babesia tubaci, commonly called as silver leaf white fly. This particular white flies carrying virus can infect tomatoes. They can also infect or this particular vectors can also transmit this particular virus to other solanaceous members. So the solanaceous members, other solanaceous member can also be the reservoir of this particular virus, and this particular vector can transmit this particular virus to their offsprings also, and this particular virus is transmitted even from one fly to the another fly, that is through it is sexually transmitted from one flies to other flies. So this is about the mode of transmission of this virus. Kindly remember the insect which transmit this particular disease. Now, we studied the morphology of the virus. We study the transmission of the virus, how actually this virus has been transmitted from one plant to another plant. You know, this particular virus is being transmitted through whiteflies. So now, let us study how actually this particular virus replicates inside the plant cell. How actually it replicates inside the plant cell. Let us study in detail. So, as I was mentioning that whiteflies are the vectors, or these whiteflies are the carriers of this Gemini virus, that is the virus which cause tomato leaf curl disease. So how actually this particular virus enters inside the plant cell, how actually they replicates inside the plant cell, let us study in detail. So let us study about the life cycle of this Gemini virus. So as I had mentioned, the whiteflies are the one which sucks the cell sap of the tomato plant, for example, cell sap of the tomato plant using its mouth part. If the plant has been infected, along with the cell sap, this particular insect also sucks the virus. Once such whiteflies enters or it come in contact with the healthy plant of tomatoes, what happens? Once it start sucking the cell sap of a healthy leaf, what happens? Through its mouth part or even through its saliva, the virus can enter inside the host cell. So what happened later? So here, you know, this particular virus is made up of what? Protein capsid. Inside this protein capsid, there is single-stranded circular DNA. So here, the entire virus may not enter the host cell. Uncoating happen. What is the meaning of uncoating? That is, the proteinaceous covering or proteinaceous sheath or the capsid is uncoated, so that only the single-stranded DNA enters inside the host cytoplasm. So for example, if this is the cell of a tomato plant, one cell of a tomato plant. So once the insect starts sucking or biting, what happens? Along with its saliva, the virus comes out. What happened? The protein capsid gets uncoated. Only the single-stranded DNA enters into the cytoplasm of this particular cell of a plant. Later, what happened? This particular single-stranded circular DNA immediately imported into the nucleus. This is the cell wall, cell membrane, this is the cytoplasm, and this is the nucleus, and these are nuclear pores. So immediately this single-stranded DNA is imported into the nucleus of this particular host cell. There what happened? The single-stranded DNA, using the proteins which are present in this particular nucleus, they undergo a process of replication and they form double-stranded DNA is being synthesized. This double-stranded DNA is called as replicative form, RF form of DNA, replicative form. Single-stranded DNA gets converted or gets replicates and forms a double-stranded DNA, utilizing all the proteins or enzymes which are required for its synthesis. Using the host enzymes and proteins, it start replicating. Later, this double-stranded DNA undergoes a process of transcription. They undergo a process of transcription. You know what is transcription? What actually is transcription? That is the synthesis of the mRNA. You know, during the process of protein synthesis, there are various steps. What are those? Transcription, translation, and termination. These are the different different steps which are involved in the process of protein synthesis. So the double-stranded DNA or the replicative form of the DNA undergoes a process of transcription. The product of the transcription is what? mRNA. mRNA is being synthesized. This mRNA, once it has been synthesized, it is being exported to the cytoplasm of that of a host. It is being exported to the cytoplasm of that of a host. Later, this mRNA, the next process of protein synthesis is translation. Translation happens, which results in the formation of various proteins which are required for the process of the synthesis of the viral DNA or a viral particles. So once again, I will repeat, once the single-stranded DNA enters inside the host nucleus, utilizing all the enzymes and all the proteins which are required for its synthesis or for the process of replication, it replicates and forms double-stranded DNA. This double-stranded DNA undergo a process of transcription. As a product of transcription, mRNA, viral mRNAs are synthesized, where they are exported onto the cytoplasm. In cytoplasm, what happens? The translations is happening. As a product of translation, you could expect the formation of various proteins. All these proteins are essential for the process of the replication of this viral genome. Once the proteins has been synthesized, again it is transported into the nucleus. There inside the nucleus, the replication of this double-stranded DNA happens by rolling circle mechanism, which results in the formation of many replicas of single-stranded DNA. These single-stranded DNA enters into the cytoplasm with the involvement of movement proteins. These movement proteins have been involved in the process of the transportation of the single-stranded DNA, which has been synthesized in the infected cells into the neighboring cells through plasmodesmata. What is this plasmodesmata? This is an opening or the connection between one cell to another cell. So through this plasmodesmata, these viral particles which has been synthesized in the infected cell gets transported to the neighboring cells, and from that neighboring cells again the infection happens. Again it gets transmitted to the another cells. So the entire plant gets infected with this particular virus. So this is about the life cycle of Gemini virus. So now, what are the disease symptom of this particular disease? So till now, students, we studied about the morphology, classification of this virus, morphology of the virus. We studied the mode of transmission of this particular virus, and also we studied about the replication or the life cycle of this particular virus. And now let us study about the disease symptom. So because of the infection of this particular virus, how actually the symptom of this particular disease can be seen externally. How actually we can see what actually happens to that of a plants because of the infection with this particular virus. So what happens once the infection has happened? The newly grown plants shows curling of the leaves. The leaves gets curled. The newly formed leaves, see for example here you could see the newly formed leaves shows curling, and also there will be a reduction in here also. This is one of a plant which has been completely infected. where the leaves have been curled, and also there is a reduction in the size. See, for example, if this is the normal leaf, this is the normal leaves. If you compare these two leaves, you can see there is a reduction in the size of the leaf. And another important thing is the internode difference. In my last class also I told what is node and what is internode. The node from the distance between the the distance between the two node is called as internode. This is internode. This is node. From the nodal region, the branches or the leaf arises. The distance between the two nodes are called as internode. So what happens here? The internodal distance become less. There is a reduction in the size of the leaf. There is a reduction in the internodal distance, and because of the reduction in internodal distance, what happens to the plant? The plant shows stunted growth, or they show dwarfism. That is, they are stunted growth. And also the newly formed leaves, as I had mentioned, reduced in its size, and it showed wrinkling. The wrinkling of the leaves happens. You could see over here, the leaves have been wrinkled, and it is showing fluorosis. That is, the leaves are becoming yellowish. They are becoming yellowish because of the lack of chlorophyll pigment. Why this chlorophyll pigment has been lost? Because all the mechanism, all the machineries of this particular cell of the tomato has been utilized by that of a virus particle for synthesizing its own DNA. That is the reason even the chlorophyll pigmentation production also hampers, and which results in the chlorosis of the leaves, especially it is being yellowed between the veins. Say for example, if these are veins, in between veins you can see the chlorosis, isn't it? That is called as in between veins. Say for example, this is midrib, these are veins. In between veins you can see the chlorosis. And also the margin curls upward. Say for example, this is the margin of the leaf. It curls upward. It curls upward and give cup like appearance. The leaves are becoming, here also you can see the leaves, the margin is raised or curled upward. Because of this, what is happening? The leaf is becoming cup-like, and also such plants may not flower. If the flower formation happens also, the fruits are not formed, because by the time the pollination happen, fertilization happen, the fruit production happen, by that time the flowers itself would have just dropped down. So these are, because of this, what's happened to the economy of the production of this particular tomato may not happen. This entire plant may not produce tomatoes at all. So these are some of the disease symptoms. It is curling of the leaf, reduction in the size of the leaves, chlorosis, internodal distance reduced, stunted growth, and the early fall of the flowers, and the fruit production may not happen. So these are some of the disease symptom of this particular disease. Now, how to manage this particular disease? How we can manage, or how we can control this particular disease? As we had mentioned, always prevention is better than cure. That is the reason a proper sanitary measures has to be followed. If you find any infected plants or white flies infecting the leaves and all, you take out those particular plant or the leaf or the branch, whatever it is, it can be taken out and it can be discarded. Better, it can be burnt into ashes. And avoid solanaceous members. The weeds, for example, weeds, if along with the tomato plants, if you see any other weeds, especially if it belongs to solanaceous, better always weeds has to be removed, because the competition may develop, and also the chances of infection with other pathogen can also happen. That is the reason always weeds, unwanted plants has to be removed. If there are any solanaceous members, it has to be removed, because those solanaceous members may serve as a reservoirs. As I had mentioned, they serve as a reservoir of this particular virus. That is the reason better to remove this particular weeds has to be removed, and use of UV reflective mulches. So for example, this is one of the image where they have used UV reflective mulches. UV reflective mulches. See what happens? The trap. This is one of the mechanism wherein which the flies may may not get attracted to that of this particular plant. So the UV reflective rays, no, they may inhibit the insect to attract to come towards the tougher plants. That is the reason this particular UV reflective mulches is being very much effective, and various research has also happened nowadays. In most of the plantation, they are using this kind of UV reflective mulches in order to avoid this particular insects and coming infecting the plants. And also we can use some trap crops. What is the meaning of this trap crops? Say for example, trap crops, if you are going to plant some chrysanthemum or certain cowpeas or soya beans or groundnuts or even marigold along with this particular tomato plants, the main reason behind this planting this kind of plant is to, especially this marigold or chrysanthemum, they attract the insects to themselves, so that the chances of a whitefly entering or getting towards the tomato plant can be inhibited. That is why we can use certain trap crops along with tomato growing. And also usage of this yellow sticky traps. This is one of the image of yellow sticky trap. So here, this because of this color, yellow color, the insects tends to move towards this kind of traps. So if you are going to smear some kind of grease or something, what happened to this particular color, insects will come and land on them, so that because of the grease which we have smeared on this particular yellow sticky trap, they may get attached, or they may not able to, they just die there only. So that is the reason we can have such kind of yellow sticky traps, at least 10 to 12 yellow sticky traps you can keep in 1 hectare. For 1 hectare, at least 10 to 12 yellow sticky traps. The main purpose is to attract the insect, and also if you are applying grease, the insects land over there and they stick and they die there only. So we can use such kind of methods to control this particular disease. And another method is, as I was mentioning, you destroy the infected plant or the infected part of the plant, you just destroy them, and use of certain natural enemies. Say for example, onto this white spiders, you can see the spiders as well as merit bugs, they feed on this particular insects. So better to use them, and also using of certain phytochemicals, that is plant extract, that is peripherinoid basis insecticides, especially it is made up of usually chrysanthemum and even marigold. They can, the extract can be used to check this particular insect, and better to go for disease resistant varieties can also be used. And also there are various research which tells that neem oil or the extract of neem leaf along with the soaps, and spraying with such neem oil or with the detergent or the soap may also avoid the disease. Always better to go for disease resistant varieties. Various disease resistant varieties towards this viruses are available. We can also use this to overcome this